Hello everyone. In continuation to the previous lecture on mechanisms of cell injury, we'll be discussing free radical induced cell injury in today's session. It is one of the most important and confusing topic and I try to make it as simple as possible. Travel with me in today's session on journey of free radicals and answer the questions in the end by commenting in the comment box. First, let us have a brief look of mechanisms of cell injury which we have learned in the previous session. So, the various types are ATP depletion, mitochondrial damage, increased cytosolic calcium, membrane damage, DNA damage, and the final thing is free radical induced cell injury. Today we will be discussing about free radicals in these headings: properties of free radicals, types of free radicals, sources of reactive oxygen species, mechanism of formation of various free radicals, antioxidants, effects of various reactive oxygen species, what is oxidative stress, and conditions with increased reactive oxygen species. Coming to definition of free radicals. What are free radicals? They are unstable chemical compounds with a single unpaired electron in the outermost orbit. As you can see here, there are two orbits. In the innermost orbit, you have two electrons, and in the outermost orbit, there is only one electron, that too unpaired electron. So, this unpaired electron is highly unstable and it is responsible for the high reactiveness of the free radicals and they are rapidly destroyed. Because of this unpaired electron, these free radicals initiate autocatalytic reactions whereby the molecules with which they react are themselves converted into free radicals to propagate the chain of damage. So they initiate autocatalytic reactions. It's confusing. Let me give with example. So here is a reactive oxygen species. So what is it having? It is having single unpaired electron. Now it reacts with the normal molecule, adjacent normal molecule, and tries to steal electrons from the normal molecule, thus converting this normal molecule into another free radical. And this process continues. This again converted free radical attacks the normal molecule, again it converts into another free radical. In this process, what happens is there is accumulation of more and more free radicals, thus causing oxidative stress. So these unpaired electrons, as I told you already, they are highly reactive and attack proteins, lipids, carbohydrates, nucleic acids, which damages the cell membranes and nucleus. So here this is a single unpaired electron which attacks the lipids and proteins, causing membrane damage, and it attacks the nucleic acids, causing DNA damage in the nucleus. So here you can see the unpaired electron, how it is attacking lipids, proteins, and nucleic acids, causing damage to the plasma membrane and the nucleus. What are the types of free radicals? Free radicals are of three types. They are oxygen-derived free radicals, nitrogen-derived free radicals, and drug and chemical free radicals. Oxygen-derived free radicals are also known as reactive oxygen species. In this, we have superoxide anion, hydrogen peroxide, and hydroxyl ions. In the nitrogen-derived free radicals, we have nitric oxide, peroxynitrite anion, nitrogen dioxide, nitrogen trioxide. And in the drug and chemical free radicals, we have carbon tetrachloride and CCl3. What are the various sources of reactive oxygen species formation? First and most important thing is mitochondrial respiration. In the process of oxidative phosphorylation, which occurs in the inner mitochondrial membrane, numerous free radicals are formed as intermediates. Second important mechanism is inflammation. In this process, in phagosomes and phagolysosomes, to kill microbes, numerous ROAs are formed. NADPH oxidase helps in the conversion of oxygen into superoxide anion. Enzymatic reactions are also involved in the formation of reactive oxygen species. UV radiation and ionizing radiation helps in the generation of reactive oxygen species, which causes damage to the DNA by double-stranded and single-stranded DNA breaks. Next mechanism is heavy metals and certain xenobiotics are also involved in the formation of reactive oxygen species. And the last final thing, important thing is reperfusion injury, which is also involved in the generation of reactive oxygen species. How these free radicals are generated? There are various mechanisms, and as I told you, the first and foremost important mechanism is by oxidation reduction reactions, which happens in the mitochondria. So in the inner mitochondrial membrane, what happens is ATP, that is energy, is generated by reducing oxygen to water. This is known as oxidative phosphorylation, and in this process various intermediates are formed, which are because of partial reduction of reactive oxygen forms. So here we can see this oxygen is reduced to superoxide anion by the presence of enzyme known as xanthine oxidase or cytochrome P450. And this superoxide is further converted to another reactive oxygen species known as H2O2 or hydrogen peroxide in the presence of enzyme known as superoxide dismutase. This autocatalytic reaction doesn't stop here also. It is this this hydrogen peroxide is further converted into hydroxyl ion, and this process is mediated by iron, that is Fe2 plus is converted into Fe3 plus, that is Fe2 plus is converted into Fe3 plus. In this process hydroxyl ions are formed, and this reaction is known as Fenton's reaction. And the second mechanism is inflammation. In inflammation, there is formation of phagosome or phagolysosome by neutrophils and macrophages. So these neutrophils and macrophages are going to attack the microbes and kill the microbes. How they are doing that? By means of enzyme NEDPH oxidase. Here oxygen is converted into reactive oxygen species, that is superoxide anion, and in that process there is accumulation of the reactive oxygen species and release outside, that is known as respiratory burst. So during phagocytosis, this ROAs are produced in the phagosomes and phagolysosomes, that is mainly neutrophils and macrophages, which is very similar to the mitochondrial respiration, that is accumulation of superoxide anions resulting in respiratory burst or oxidative burst. So here this oxygen is converted into superoxide ion by means of enzyme known as NADPH oxidase or phagocyte oxidase. This superoxide anion is again converted into hydrogen peroxide by superoxide dismutase, and this hydrogen peroxide is further converted into HOCl, that is hypochlorous acid, in the presence of enzyme known as myeloperoxidase enzyme, which is present in the neutrophils, and this HOCl is highly reactive and thus helps in killing the microbes. Because of the defect in the NADPH oxidase, there is decrease in the superoxide anions production, leading to ineffective phagocytosis, which leads to a disease known as chronic granulomatous disease. Third mechanism of formation of reactive oxygen species is by transition metals. Transition metals like iron and copper, they are able to donate or accept free electrons during intracellular reactions and thereby catalyze free radical formation, as in the Fenton reaction. As we have already seen, H2O2 or hydrogen peroxide is converted into hydroxyl ion. In this process, Fe2 plus, that is ferrous form, is converted into ferric form. So this is known as Fenton reaction. And the fourth mechanism is enzymatic metabolism of chemicals and drugs. Certain chemicals like carbon tetrachloride, they are able to generate free radicals which are not reactive oxygen species but have similar effect similar to them. And example is carbon tetrachloride. It can generate CCl3, which is a free radical and causing damage to the liver. Fifth mechanism of formation of reactive oxygen species is by radiation. UV rays and ionizing radiation, it can hydrolyze water into hydroxyl ion, and this hydroxyl ion, as I told you already, it is highly reactive free radical. So here you can see how this water is hydrolyzed into hydroxyl free radical. And the sixth mechanism is from nitric oxide. What is nitric oxide? It is a chemical mediator of inflammation. It is generated by the endothelial cells, macrophages and neurons. It also acts as a free radical and it gets converted into highly reactive nitrogen species like peroxynitrite anion, NO2, that is nitrogen dioxide, and nitrogen trioxide. How is it formed? From the superoxide anion, when this combines with nitric oxide, it gets converted into peroxynitrite anion in the presence of nitric oxide synthase. So free radicals are produced within our body itself, but our body is able to escape the free radical induced cell injury by certain substances known as antioxidants. Free radicals are inherently unstable and generally decay spontaneously. So what are the antioxidants? There are enzymatic antioxidants and non-enzymatic antioxidant. What is an antioxidant? First of all, antioxidants are the substances which binds to the free radicals and neutralize them. So in the enzymatic antioxidants, we have superoxide dismutase, which neutralizes the superoxide anion and converts it into hydrogen peroxide. Catalase, which is present in the peroxisomes and neutralizes the hydrogen peroxide into water molecules. Glutathione peroxidase, which helps in the neutralization of hydrogen peroxide into water molecule, and in this process reduced glutathione is converted into oxidized glutathione. Coming to non-enzymatic antioxidants, we have exogenous sources and endogenous sources. In the exogenous sources, we have vitamin A, vitamin E, and ascorbic acid or vitamin C. Sulfitol containing compounds like cysteine and glutathione are also examples of exogenous non-enzymatic antioxidants. Coming to the endogenous sources like transferrin, which is iron binding transport protein, ferritin, which is storage form of iron, and celluloplasmin, which is copper binding protein. So this transferrin ferritin, it binds to the iron and makes unavailability of iron for Fenton reaction, so that free radicals are not formed from the transition metal. And celluloplasmin, it binds the excess of copper, so that copper is not available for free radical generation. In this process, so there is excess of removal of iron and copper, thereby preventing hemochromatosis, which is because of iron overload, and Wilson's disease because of excess of copper overload. Coming to the types of superoxide dismutases and their role in amyotrophic lateral sclerosis. Generally, what is happening? Superoxide anion is converting into hydrogen peroxide in the presence of superoxide dismutase. These superoxide dismutases are of two types: superoxide dismutase SOD1 and SOD2. SOD1 is copper zinc superoxide dismutase, which is present in the cytoplasm, and SOD2 or manganese superoxide dismutase is present in the mitochondria. So C for C, copper zinc is in the cytoplasm, manganese M for M in the mitochondria. What is the specialty of this superoxide dismutase is, it is an enzyme which protects the brain from free radical injury. So when there is mutation in the SOD1, that is copper zinc superoxide dismutase, there is absence of copper zinc superoxide dismutase. So what is happening? There is accumulation of free radicals, that is superoxide anions, within the brain, resulting in diseased motor neurons, causing amyotrophic lateral sclerosis. So what is the mechanism of amyotrophic lateral sclerosis? That too familiarly is because of SOD1 mutation. Coming to free radicals generation and neutralization, superoxide anion is getting converted into hydrogen peroxide in the presence of superoxide dismutase. So superoxide dismutase is an enzymatic antioxidant which helps in the neutralization of superoxide anion. Now the hydrogen peroxide which is formed gets converted into hydroxyl ions. In this process, Fe2 plus is converted into Fe3 plus, and this process is known as Fenton reaction. The hydroxyl ions which are getting formed by this process are highly reactive, and cell is incapable of neutralizing these hydroxyl ions because they are highly reactive, hence react with the adjacent molecules spontaneously. But there are neutralization mechanisms for the hydrogen peroxide. There are two mechanisms. First mechanism is it gets neutralized by enzyme catalase and forms into water molecule, and this process occurs in the peroxisomes. And the second mechanism is it gets converted into water molecules by the presence of glutathione peroxidase, in which reduced glutathione is converted into oxidized glutathione. So this mechanism is mediated by glutathione peroxidase. This glutathione, the ratio of reduced glutathione and oxidized glutathione, it indicates oxidative stress present in the body. So the intracellular ratio of oxidized glutathione to reduced glutathione reflects the oxidative stress of the cell, and it is an important indicator of cellular redox balance. Coming to the pathological effects of free radicals. These effects are because of three mechanisms, that is lipid peroxidation, oxidative modification of proteins, and lesions in DNA. Coming to lipid peroxidation first, what happens is the same mechanism which I told you in the starting. There are unpaired electrons in the outermost orbit of all the free radicals. So these unpaired electrons are going to react with the membranes, and in the membranes what are present? Lipids. So this unpaired electron tries to steal electrons from the lipids, and in that process it converts them into free radicals, that is lipid peroxides are formed, and this lipid peroxides cause membrane damage, finally leading to plasma membrane damage. So how is this happening? Double bonds in unsaturated fatty acids of membrane lipids are attacked by reactive oxygen species, especially hydroxyl free radicals. So as I told you, hydroxyl free radicals are the most potent free radicals. It forms into lipid peroxides, and now this lipid peroxides which are formed are unstable and react and initiate autocatalytic chain reaction, which I told you in the starting of the session, which causes further membrane damage. Coming to the second mechanism of damage, that is oxidative modification of the proteins. These reactive oxygen species, so as I told you, it has unpaired electron, it reacts with the proteins and causes various modifications in the proteins. That is, it causes oxidation of the amino acid side chains and formation of covalent protein-protein crosslinks, especially disulfide bonds are attacked, and there is oxidation of the protein backbone, damaging active sites of the enzymes, and it enhances the formation of unfolded or misfolded proteins, and these misfolded proteins or unfolded proteins are further degraded by proteasomal mechanism. And coming to the lesions in the DNA, how these reactive oxygen species are causing lesions in the DNA. So ROS causes single and double stranded breaks in the DNA. There is cross linking of DNA strands, and it helps in the formation of adduct formation, that is the molecules they react with the reactive oxygen species and forms adduct. And what is the role of this oxidative DNA damage? It has role in cell aging and cancers, cell injury and death by necrosis and apoptosis. So coming to oxidative stress. What is oxidative stress? Oxidative stress is the result of imbalance between reactive oxygen species and antioxidants causing increased ROS. So ROS are trying to damage membrane, nucleic acids and proteins, whereas antioxidants are trying to counteract the effect of ROS. So there is a balance in between ROS and antioxidants so that we are in a stable state. But in oxidative stress, what happens? There is increased production of ROS, so that this antioxidants are unable to neutralize all the ROS, that is reactive oxygen species. So in that process, reactive oxygen species have upper hand over the antioxidants. So this leads in the accumulation of more and more free radicals, causing damage to the membranes, nucleus and proteins. So oxidative stress, it has been implicated in a wide variety of pathological processes, including cell injury, cancer, aging, and some degenerative diseases such as Alzheimer's disease. This you can see in its diagram. So oxidative stress causing aging, degenerative diseases like Alzheimer's disease, and cell injury. What are the various pathological conditions associated with free radical induced cell injury? It has a role in cancer, atherosclerosis, myocardial infarction, stroke, Alzheimer's disease, diabetes, cataract, inflammatory diseases and aging. And lot of research has been is being done on reactive oxygen species, their role in various mechanisms. So this is the gist of reactive oxygen species. So there are endogenous source, exogenous source, ultimately leading to free radical formation, which can be of reactive oxygen species and reactive nitrogen species, ultimately leading to cell damage by DNA damage, protein oxidation and lipid peroxidation. These are the questions framed from today's topic. You can solve these questions and give your answers in the comment box. Hope this session has cleared so many doubts and confusion on free radicals. If you feel this video has been useful to you, I request you to like, share and subscribe. Thank you.